Okay, let's go to the next one where we show. So this is more detail, and some of this detail is really important. Some of this, if you kind of commit to learning how it works, you'll have to memorize less. So again, I would encourage you go back and forth between details like this and big picture, like where these lipoproteins are going. So if we look at this, some similarities we saw before, phospholipids on the outside. So this is the level of detail I'd want you to draw. You'd want to be able to draw one of these. And you can put phospholipids on the outside. Here's cholesterol. What does that little black circle mean? That's the hydroxyl group, right? It's a little bit polar. That puts it in that position on the outside of that lipoprotein. How's that different than this? Fatty acids attached. All right? So the only polar component of that cholesterol has now been replaced with a fatty acid. So now it's completely hydrophobic. It goes inside. So when you draw these and I say include the molecules in the core, you're going to put cholesterol ester. On the surface is cholesterol. And I'll tell you why that's important coming up. Okay, what else is here? Triglycerides. All right, glycerol backbone, 3 fatty acids. That's what these are. And a couple other things here are apolipoproteins. Again, they should. I should figure out how to fix this. Peripheral apolipoprotein on the outside, loosely associated with the outside. That's different than this one. See this? This is kind of an integral apolipoprotein. See the way it's wrapped around and kind of embedded in that layer? That's either apolipoprotein B100 or apolipoprotein B48. There's a lot of nomenclature on these. And again, maybe kind of a boring crew that came up with all these. This is just based on protein size. B48, B100. So guess how big B48 is? 48% the size of B100. Right? And we'll talk about this in 329 lab. These actually come from the same gene. So it's a very interesting process, and it has to do with our risk of cardiovascular disease in people is related to that gene. So that's some of the genetic ramifications involve B100, B48, that gene, ApoB gene. But look over here. As a matter of fact, you can cross this one off, ApoE, apolipoprotein E. Have you ever heard of that one? There's like some studies that indicate different forms of apolipoprotein E are related to Alzheimer's disease. We won't go into that here, but these things are not just for simply moving lipids in the blood. They have significant health impact on some of these. And there's lots of subtypes of these. It drives you nuts. Okay, there's C2, C3, C3, C4. I think it goes all the way to C8. There's lots of these. Anyway, we're just looking at A1 and C2. Now, so they're on the outside. ApoA1, C2 loosely associated with the outside. B48, B100, integral, large, embedded apolipoprotein. That's what they look like. I switched to this slide not too long ago. I had a more detailed one, and this one looks a little bit more warm and fuzzy. But it's got 3 different parts to it. And this is actually from, I think, Leninger Biochemistry, which is probably my favorite version of biochemistry textbook. Okay? It looks messy. So what I would encourage you to do is memorize this or learn this in 3 parts. So part number 1 is going to be this. And when we do it this way, you'll see it's relatively simple. So this is kind of what we call overall. This diagram I would name lipoprotein metabolism. When you are going through this, practice drawing it. Practice drawing the three parts. And we're going to add more detail to this. So you can write something, draw something that resembles the gut. Remember, chylomicrons are made in enterocytes. And then you can see chylomicrons here, they go into capillaries. There's a capillary, but what are we missing? The lymph. So make sure you put on your diagram or something, chylomicrons are made in enterocytes, they enter the lymph. And then the blood. What do they have inside? Mostly triglycerides. From where? The diet. What's the other lipid they have inside? Cholesterol esters. Where did it come from? The diet. Consume a meal, it's got triglyceride cholesterol. It ends up in chylomicrons. Chylomicrons enter the lymph, then the blood, and then look what they do. Then they go through capillaries and tissues. So the example tissues, mammary tissue, muscle tissue, adipose tissue. And now look what's happening. As the chylomicrons move through those capillaries, they lose their free fatty acids because they are hydrolyzed by protein lipase. So you got chylomicrons there. Let me draw this. Try to do this quickly. I'm going to have to get rid of some of the writing here. So this is a capillary. So here's the blood. These cells are making up the walls of the capillaries. What do we call these cells? Endothelial cells are the dominant cell type that make up the walls of blood vessels, endothelial cells. These are the ones that are involved in cardiovascular disease. Endothelial cells that line the blood vessels within the heart. When those endothelial cells become affected in certain ways, it can lead to the progression of plaque. Okay? But they're also in the other tissues as well. On the outside of these, I'm going to put this, equals a goalpost. Lipoprotein lipase. And this is what lipoprotein lipase does. Hydrolyzes triglycerides to fatty acids plus what? Glycerol. So here's a chylomicron. Lipoprotein lipase is on the surface of the endothelial cells. It binds to chylomicrons. How does it bind? ApoC2. Chylomicrons also have, no, sorry, let me jump ahead. This table is a good summary. Shows chylomicrons deliver fatty acids attached to triglyceride derived from dietary fats and non-hepatic tissues like muscle and adipose tissue. Chylomicron remnants now deliver dietary cholesterol to the liver. So those first two lines are important for this. The next slide talks about apolipoproteins and their functions. One of the functions is structural component. For example, the synthesis of chylomicrons relies on ApoB48. They provide recognition sites for cell surface receptors. We'll talk about that for LDL. The last ones, they act as cofactors for enzymes involved in their metabolism, specifically ApoC2, that activates LPL. So that's what's happening here. ApoC2 is on that lipoprotein, so it can bind and activate lipoprotein lipase. So that the triglycerides can be hydrolyzed, so that the fatty acids do this. Have you ever talked about this in physiology? We talk about structure of tissue. You've got endothelial cells that are making the capillaries. Some molecules can pass through or in between those endothelial cells. So nutrients and even things like fatty acids can leave the capillary, the lumen of the capillary, go in between the cells and end up in the underlying cell type. So for example, this is likely a fat cell. And in those fat cells, they're reassembled. So when I eat dietary lipid and it ends up in chylomicrons, those chylomicrons are going to go through the blood, they're going to go through capillaries and fat tissue, and they're going to be hydrolyzed. The fatty acids go into the fat cells, reassembled into triglycerides. So essentially what have I done? I've stored that major energy source that I consumed. So now the trick is, if I don't want to retain that fat, later on, I hydrolyze the fat, physical activity and other things that will do that, and now it reverses. The fatty acids go into the blood, they're used for energy. So the main point of this is chylomicrons deliver triglycerides to extrahepatic tissue. So out of these three tissues, it's more likely to end up in adipose tissue. And it has to do with the KMs of different forms of lipoprotein lipase, but that's where most of it goes. So most dietary triglyceride ends up in fat tissue. Okay? So let's add a little bit more detail to this. This also has, I'm just going to draw it like this, ApoB48. That's a very large protein that wraps around a chylomicron. And years ago when we were doing research, we were one of the groups that went, how many apolipoprotein B48 molecules are there on these particles? And we found out that it's big enough, it's typically one ApoB48 per chylomicron. It can wrap around that big thing. And it kind of acts as a belt. It's a structural component, but it also has some other things as well. So now what have we done? We've dropped off triglycerides to tissues. Now, what's left in that chylomicron remnant? Cholesterol. So put this in your notes. C2 activates lipoprotein lipase. ApoC2 activates lipoprotein lipase, which hydrolyzes triglycerides. So fatty acids are delivered to cells. When approximately 80% of the triglycerides are removed, ApoC2 is released. The chylomicron remnants released from LPL. And travels to where? The liver. Chylomicron comes into the tissue. C2 allows it to bind to lipoprotein lipase. Triglycerides or hydrolyzed fatty acids are absorbed into the tissue. That happens until that chylomicron shrinks. When it shrinks, ApoC2 releases. It's no longer binding to LPL. So now the particle is released. Because it's been hydrolyzed, we call it a remnant. Chylomicron remnants go to the liver. Their metabolism's done. They go into the liver, and what was on their inside now mostly? Cholesterol esters. So chylomicrons deliver dietary triglycerides to extrahepatic tissue and cholesterol to the liver. That's what that table says, right?
What does B48 do again?
It's required for structure, for making that chylomicron remnant. So as those chylomicrons are made, there's protein structures like a scaffold on the surface of that ER, so that as the lipids go into the ER, it's interacting with ApoB48. So as the lipids go in there, they interact with hydrophobic regions of B48, and the whole thing kind of comes together. And then towards the end, when you get enough of it there, the ApoB48 is essentially wrapped around. You get a large-sized particle. That's an indicator that that chylomicron's done. It moves from that scaffold through the ER to the Golgi, and then it's secreted. So B48 is a scaffold for its synthesis. That's it for chylomicrons. That's step one. We have two more. The second one is, we're going to call this VLDL, LDL metabolism. And this one's hard to believe, even more fun than chylomicrons. So now look where VLDL start. They start in the liver. So VLDL contain endogenous lipids. We've talked about that before. We said, oh, if a person consumes too many calories, too many carbohydrates, some of the glucose can be converted to fat. The fat can be put into VLDL, it goes into the blood, it becomes LDL. We talked about that with fiber. We talked about that when we talked about carbohydrate metabolism. And here's where they are. So VLDL leave the liver. They have ApoB100. So on here, when you draw this, you could put like, here you can put your B48, and you can put your, for VLDL, you can put B100. And B100 is even bigger than B48. So it's wrapped around that VLDL molecule. When VLDL go into the blood, they pick up ApoC2. Why do they need ApoC2? Because look at their metabolism. They do the same thing. VLDL travel through the blood, enter capillaries, bind LPL. Their triglycerides are hydrolyzed. Triglycerides are dropped off. Again, what's the liver trying to do? Get rid of triglycerides. Liver's not a storage place for triglycerides, so it will purge them in VLDL. Best place to store fat? Fat. And now this is basically the same. So what would we have to change here? Change this to B100. Still mostly triglyceride, cholesterol ester. If you change this to B100, this is VLDL. Size, density, but yeah, drawing it like this. And then what does it become? This happens too. Triglycerides are released, particle shrinks, C2 is released. But now rather than chylomicron remnant, what do we call it? VLDL remnant. That's the easiest one to remember. It's also called IDL. And that's why this says IDL is intermediate density lipoproteins, because we don't have enough names. So this is intermediate density lipoprotein. But here's where it's different. VLDL remnants can be cleared by the liver or become LDL, the dark side, right? Or they can go to LDL. So what do LDL have mostly? What lipid? Cholesterol. Why? Because they were VLDL, but they lost most of their triglycerides, so they are mostly cholesterol. So on your diagram, so one of the questions is, well, how much does this? How much goes to the liver? How much becomes LDL? About half. So on this diagram, on this fork in the road, 50%, 50%. 50% of VLDL remnants cleared by the liver. 50% become LDL. Okay? So essentially, what do VLDL remnants do? They deliver cholesterol back to the liver. Or they become LDL. You got another branch in the road, a fork in the road here. Here's LDL. Look what they can do. They can either go to extrahepatic tissue or they can be cleared by the liver. So the percentage here is 75% of LDL are cleared by the liver. 25% are taken up by extrahepatic tissue. And why do we call them bad? Because extrahepatic tissue includes coronary arteries. And that's when it's problematic. That's when it's not performing its optimal function. That's kind of an artifact in this process. Okay? So LDL, absolutely essential, critical for life, but can be problematic. And so think about what's happening here. The liver, grand central station for metabolism. The liver has the ability to make lots of molecules, lots of types of molecules, including cholesterol. So in our body, the liver makes most of the cholesterol. It can make a lot more cholesterol than other tissues. So what does it do? It supports cholesterol metabolism of other tissues. And how does its cholesterol go from the liver to extrahepatic tissues? LDL. So it's crucial. Gotta have it. Every cell needs cholesterol. If it can't make enough, it can pull it from the blood. So what we're going to learn is we can actually manipulate diet and lifestyle to modify how much LDL goes this way and how much goes this way. Remember we talked about fiber before? Fiber's involved in all of this and some other things. So we'll talk about all that. But that's basically VLDL, LDL metabolism. And we'll look at a little bit more specifics in a second. Yep.
Is that all happening in blood capillaries? Or like, where is that happening?
This part is in capillaries, but it's occurring in the major vessels as well, like LDL and VLDL. It's going in between in the capillaries, and then it'll go back into larger circulatory system, larger vessels until it hits main arteries or veins, and then so kind of everywhere. But the actual conversion from VLDL to VLDL remnants is in the capillaries. Did I miss anything on that? Okay, last one, right? 3. Maybe your favorite one. HDL. Now, I think if we look at this, I didn't mention this before, but look. HDL can actually be made in intestinal cells. So HDL tend to be made in intestinal cells and liver cells. That's where a lot of them are made. We'll talk about that coming up, but we'll talk about how they function in a second. But oh, actually, this is probably a good one. This is what HDL does: collects or scavenges. Have you ever heard that term? HDL is a cholesterol scavenger. It picks up cholesterol from non-hepatic tissues and delivers it to the liver for excretion. So overall, that's a good thing. It's not great for Sean and I because we tend to have lower HDL than everyone else in here, right? So HDL is considered to be protective. And in general, women tend to have higher HDL cholesterol levels than men. And that's potentially one of the reasons the lifespan of women is longer. It's protective. It's potentially one of the reasons. So you'll see, like in 329 lab, if you want to do it, you'll measure your total cholesterol, your HDL, your LDL. And I did the averages this quarter, and overall you can see the HDL cholesterol values in the Cal Poly nutrition program pretty high. Pretty high, right? And so overall, that's what's happening is now if you take a blood sample and you say, oh, that's cholesterol in my HDL, where's it going? To the liver for excretion. That's why we say, oh, that's a good cholesterol. It's going that way. LDL, going the other way. Otherwise, cholesterol is cholesterol. Just kind of depends on what particle it's in and where is it going. How do these lipoproteins kind of know where they're going? They're apolipoproteins. That's what's driving their metabolism. So we'll talk about that in a second. So this one's pretty simple. HDL are made and then can pick up cholesterol from extrahepatic tissues and take it to the liver for likely excretion. And how can we get rid of that cholesterol? In what? In the feces, but where did it come from? How did it go from the liver to the feces? Bile. So some of that cholesterol can go in as cholesterol, but some of it can be converted to bile acids. And then it's no longer cholesterol, and so it decreases blood cholesterol levels. So we're going to talk about some of the things that affect the levels of LDL and HDL, and then some of the health implications of that. All right? I think, oh, and this is another good one. God, so many good tables. All right. This one, for the molecular mass, you can get rid of this. I don't want to ask you that. Fascinating, but I want to ask you this. ApoB100 is one of the largest proteins in the body. There's a handful of proteins that are bigger. Titin's one of them. There's a couple other ones, but B100 is massive. And that has some issues. But here's what they do. ApoA1, you can see the function. Some of these won't make sense now, but B48 does, right? It's synthesis, structural molecule. B100, look what it does. It binds a receptor. And it's that interaction that includes genetics and risk of cardiovascular disease that we'll talk about. And C2 is the activator for LPO. That's why you have to have that there. So, and this is a summary. So as you're drawing out your diagram, plug in your apolipoproteins. Draw chylomicrons and go, okay, they have B48 and C2. HDL, you're going to put A1. ApoA1. Right, see down at the bottom. Has some C2, but A1 is the crucial one. LDL, the only apolipoprotein is B100. It loses C2, right? It comes off. And then VLDL, we say V100, C2. I have APO. I took ApoE off of here, but ApoE kind of floats around. So here's some text that describes the diagram of VLDL metabolism, for example. We talked about all that. So here's what I mean. Here's a fun fact. Here's the lipolysis cascade. It's called VLDL become IDL, and then some become LDL. All LDL are from VLDL via IDL, but not all VLDL become LDL, right? So what that means is all of the LDL come from VLDL. So there's some things that we can do that control the amount of VLDL that we make. And if we control that, it affects LDL, because that's the only place LDL can come from. But some VLDL remnants cleared by the liver. So not all VLDL become LDL. All right? I'm not sure what was going on in my head when I wrote that out. Yeah. But probably, yeah, I'm not sure why I did that. Right? I think the rest of it we talked about. And this is some interesting stuff about LDL particles. So LDL, their main function, right, is to transport cholesterol made in the body to extrahepatic tissues. And we call it bad cholesterol because they can actually deposit cholesterol on coronary arterial walls. And they only have B100. Here's a difference. LDL tend to stay in the blood for 2 or 3 days. They have the longest half-life of the lipoproteins. And because of that, they can become altered in the blood. And altered LDL are a problem in general. Our body recognizes their alterations, targets them, and they're more likely to become part of our atherosclerosis. The video will talk about that. Uh-huh.
Did you say like the process? Because there was like a link to like a PowerPoint slide thing for it, but
There should be a link in the extra credit handout, I believe, right? I'll double check that. I don't think I didn't put the link.
it wasn't in like the YouTube page.
Oh, no, I didn't put it in there. Sorry, I can't. I should. Yeah, good point. The fiber one's in there. Why didn't I put that one? Yeah, good point. Okay, so the other ones, like chylomicrons, only stay in the blood for maybe a few hours, and then they clear. Obviously, right? You just fast and they go away pretty quick. And VLDL clear pretty fast. HDL a little bit longer, but the longest one is LDL. So here's the problem. The particle that can hurt us the most is the one that we have most of, and the one that is the most likely to be damaged in a way that leads to atherosclerosis. So we're what are called LDL animals. Rats, HDL animals. Not a lot of good things about being a rat, probably. Their lipoproteins, that's one good thing. They have mostly HDL. Rats don't get heart disease. They got other things to think about, right? Paying for rent, all that kind of stuff, right? But I got a lot of HDL. So you don't use those as a model for atherosclerosis. But some other animals, like hamsters, we switched when I was later on, anyway, a long time ago. We switched from rat model to hamsters because we went, well, they're more similar to us. And we pulled out some of these enzymes, and that's when cloning stuff and getting in gene bank was a big deal. You guys don't even know what that is. Well, got my name in gene bank. Everybody's like, yeah, all right. Should we go to the gym or have some other fun? But this is when you're looking at articles like for senior project or anything like that, and you're looking at atherosclerosis, you have to consider the model. If they're looking at how dietary things in animals affect atherosclerosis, it depends on the model. The model that they're using, some models are not appropriate. Rabbits, good model. Hamsters, good model. Rats, not a good model. Mice, probably not a good model. And it has to do with lipoproteins, and that's why some animals are susceptible to heart disease and some are not. So LDL is a key player in this. This table is very interesting, and especially next quarter when you do your blood work, I'll put this table up again, and you can see how your profile it compares to general numbers. So you can cross out triglycerides. Let's just focus on cholesterol. This is a distribution of triglycerides and cholesterol among fasting lipoproteins in humans. So this is a fasting blood sample. And now let's say, for example, you go in the lab and you go, hey, my plasma total cholesterol is 176. And I can tell you that's probably a normal value. This last quarter, I think it was 156 class average, which is pretty good value. Typically, it's 160 to 170, but they're VLDL. So now, have you ever seen that, right? You go, hey, what's your total? And then you look at the subfractions.
What is the percentage?
So if we say, hey, my total cholesterol in the blood, that's 100. Now, where is it? About 5% in VLDL. Why is that so low? Because it's fasting. VLDL clear pretty quickly. IDL even lower because they'll either convert to LDL or be taken up by the liver pretty quick. Look at LDL. 115. 65%. Humans carry most of their cholesterol in the blood and LDL, the particle that can hurt us. Look at HDL. 25%. And I can tell you right now, that's a decent value, not for women. Women will tend to be 50, 60, 70. Men, 46, probably happy with that. 45. Right? I've seen them as low as 21, 22. Not in this, but friends of mine that say, hey, can you look at my blood lipids? Sounds weird, huh? I don't have many friends, and the ones that I do want me to look at their, hey, yeah, maybe that's why they're my friend. But if you look at these, that's not a great value. But remember, when we see this, we're like, oh, that's protective, right? So we try to do things to increase HDL cholesterol. We'll talk about that a little bit. But that's kind of what. Now, there's some people that have very similar. They might have 70, and there's a small fraction of the population that actually have higher HDL cholesterol than LDL. And cardiologists have a term for that group. I don't remember what it is. It's something more classy than lucky. But that's kind of rare, but that can be protective. So those are the overall kind of average values. So one of the points of this is, again, we're mostly LDL cholesterol. There are lots of things that affect that, because one of the things that we're going to talk about is, how does this happen? How does LDL leave the blood and go into tissues that need it? It's a highly regulated process, and that gives us an overview of how we can affect LDL cholesterol in the body. And so we're going to learn some details about that. This is the next slide. And I'll give you an overview of this and then we'll stop because there's steps in this that we won't get through and I don't want to hurry through it. But on this diagram or in your notes somewhere, so this says plasma membrane. You can say of any cell in the body.
Like transportation. This is transportation of LDL to tissues.
Oh yeah. So sorry. Let me give this a title. I thought the crow was bad. That was probably worse. Right? Plasma membrane. So plasma membrane of nearly any cell in the body. The title of this is LDL receptor mediated endocytosis. You've heard of that before, right? This is LDL receptor mediated endocytosis. And this diagram looks kind of funky. This is actually based on the first paper where a group figured this out. And so as a little bit of background, the two most important people in cholesterol metabolism in the last 50, 75 years are two individuals called Brown and Goldstein. And one of them was from Texas. The other one was from New York. They were MDs that were working in medical clinics. They're both MDs. They were working in clinics in New York. And patients came in with extremely high blood cholesterol values. And they quickly figured out it's genetic. And they coined the term, it's familial, which is genetics, right? Hypercholesterolemia, which means what? High blood cholesterol, hypercholesterolemia. These individuals had so much cholesterol in their blood, some of it was actually kind of depositing in between tissues. So they actually had like bumps in their skin that were full of cholesterol. And obviously it compromises lifespan. And so they started trying to sort this out. They figured it out actually pretty quick. I would guess 10 or 15 years. So early in their careers, they got the Nobel Prize for this. And it's probably been 5 or 7 years ago. I saw one of them talk. I saw Goldstein. I was at a scientific conference, and you guys, I know you can relate to this. They're like rock stars of science. So anytime they gave a talk, the whole thing is absolutely filled, and it's a big thing. And I'm sitting in there, right? And this is how long ago it was. It's actually been a while ago. I'm sitting in there trying to figure out my BlackBerry. I'm looking, and I look over, I'm like, oh, I'm not the only one struggling with it. The guy over there's going, oh, this guy, bald guy, small guy. And I'm like, I didn't know who he was. It's Goldstein. I'm trying to figure out his BlackBerry. And then like, I'd like to introduce Goldstein. Goes up there and walks. Extremely sharp, extremely nice guy, obviously. And they had followed this all the way through where they came out with this, and then they were looking at every detail of this. And he kept joking around saying, and when I figure that out, I'm going to retire. He's probably 70. Like, when we figure that out, I'll retire. Well, maybe not. When we figure this out, that's when I'm going to retire. And just kept going. They were phenomenal when they figured this out. Not only does it affect familial hypercholesterolemia, but the regulation is affected by diet and drugs and other things. This led to discovery, basically the use of a class of drugs called statins. Once they figured out how this is regulated, other people started figuring out how to manipulate the regulation to help a lot of people. And so as we go through this, it's important that we understand the details of not only how this works, but how it's regulated. So that's where we'll go on all of this. And this diagram is kind of cheesy looking because it's from one of those early articles. So bear with me on that. So next time we'll go through, next time let's take an exam. And then after that, we'll go through the steps on this. All clear on this? Okay, guys, thank you very much. So I'll see you guys when? Sunday or Monday or Tuesday for sure, right? You can figure out kind of what works for you and let me know.